Hello, everybody. Thank you for being here. I'm Sandhya Prabhakaran. I'm a research fellow at Memorial Sloan Cancer Center in New York City. And this is joint work done with Julia Vogt, who is now at University of Basel and the Swiss Institute of Bioinformatics. And she'll slowly move to ETH, I think, in a month or two. So this work is a Bayesian model that allows you to cluster objects that are distances, that are present as distances. So before I jump into the model per se, I just wanted to take a step back and introduce you the two religions of machine learning as many people view it. So the first are frequentists, which are those set of people who love doing repeated experiments, and they only believe in the outcomes they see. There's no way they would accommodate an outcome that they haven't seen ever. And then you have the Bayesians, supposedly the cooler people, who love to observe, guess, experiment, make predictions, and they are the ones who like to live with uncertainty. So for example, if you take a coin toss and do 10 tosses and you get 10 heads, if you ask a frequentist what's the relative frequency for finding tails, you'll not get an answer, because for them, probability is a point estimate. Whereas for a Bayesian, probability is a distribution. So even if you don't see an observation, there's always a possibility of what the different outcomes are, and then you just run a uniform distribution across. So in the case of a coin flip, it would be 0.5. This is then something that I pulled off the net, which is a Bayesian is someone who vaguely expects a horse, catches a glimpse of a donkey, and strongly believes he has seen a mule. I myself am a Bayesian, so I really know that this rings true for me. So with Bayesian formulations, there's more flexibility. In modeling, you can make inferences, you can make predictions, there's thinking, lots of downstream analysis you can do. And my personal thought is that there's also one more religion, rather, that I would call a revolution, and that's deep learning. But we've seen many talks today and also tomorrow, so I won't go into that at this point. OK, so now, one of the most important and common things we do when you're given a data is to try to find out what's the underlying structure. And one of the tasks you do is that of clustering. So you have both Bayesian and non-Bayesian methods. Some of your Bayesian methods would be, non-Bayesian would be like k-means or hierarchical clustering, whereas Bayesian methods would be Bayesian, hierarchical clustering, or mixture models, et cetera. So what would you want to do? You want to find out sets of objects where objects within a set are similar to the ones that are not present in that set. So you would run something fancy or something very simple, and you would get them clustered, where every object has something very specific to it. So each color over here is depicting one particular cluster. Now, in general, the kinds of data that we work with, let's say they are sequence data or time series data, at the end of the day, you pose it as a count matrix. By that, what I mean is you have n observations. Each observation is d-dimensional. So you stack, if you have n different observations, you stack them as a matrix, and you get what I call here as x, n times d. And these are then vectorial objects. They're actually a small subset of the objects or different data types that you find in the entire universe, which I depict with the blue bubble. So once you have this kind of a data, you can plug in your favorite clustering method, and you get clustered. And everything is nice and clean. Now, the question we are asking is, what if you do not have an object that falls in the yellow bubble, that is, it's not vectors, such as non-vectorial objects? Examples are protein structures, chemical compounds, graphical networks, probability distributions, strings of text. So these are objects you do not have a direct vectorial embedding. Someone gives you this, and you want to cluster that. What would you do? So this is a space that this paper, this work deals with. And one of the properties of these non-vectorial objects are the pairwise distances, meaning you can always compute the pairwise distances between objects that you observe. Let's say they're probability distributions. You can always observe the pairwise distances between these, and then come up with a matrix like this that you see over here, which is known as D. So n times n is a symmetric matrix, and it follows all the properties of a symmetric matrix. And so given this, the question we are asking is, can we cluster this in a way that you assign multiple different clusters to the object, as opposed to assigning just one cluster to one object? So we are breaking from the traditional way of clustering, where you ideally would have just clustered one object, one cluster. Now you want to assign the same object to multiple clusters. and it's a Bayesian framework. So taking one more step back, what you see here is the Euclidean coordinate system, where you have two vectors, x1 and x2. And we can also assume this to be as two draws from a Gaussian distribution centered at 0. And if you draw this, if you compute the similarity matrix via x transpose, you get a matrix like this, and you can assume it to be from a central Wishart. Let's say you rotate the same matrix, coordinate system. You get two vectors, x rod and x dot 1 and 2. And if you compute the similarity matrix, you get, again, something that is shown in green here. And you can notice that the similarity matrices X are the same. So if someone gives you the similarity matrix alone, there's no way you can know what took you from X to S. You've basically lost the translational, you basically lost the rotation information. Now, if you translate the origin, and you have X star 1 and X star 2, so the ones in red that you see, and you compute the similarity matrix, you get something different. And this is now assumed to be a draw from a non-central Wishart. And I forgot to mention, a Wishart is a distribution from which you draw matrices. From a Gaussian, you draw vectors. From a Wishart, you draw matrices. And this is the formulation we're using here. But one of the interesting things to note is, once someone gives you these S matrices and you compute the distances, they're always the same. So if you only start with distances, then you've completely lost translation as well as rotational information. So we wanted to drive home this point that, and this is the more mathematical representation, which says that if you start from x's, the ones in green, and you move to s, where you lose all your rotational information, and then from there you go to d via this equation here. It's a unique mapping, but when you move back from d to s and s to x, that's the dotted lines, it's a non-unique mapping. So you definitely do not want to embed something that is given to you in the distance space. So we want to just work with distances per se. And how do we do this? It's a Bayesian model. You're given D to start with, and you're interested in inferring a Z matrix, which is the cluster assignment matrix, and this is how it looks like. It's a binary matrix. Every row is an object of interest, and every column is a cluster. This is what you want to infer, and I'll come to it in a few more slides. Given that this is a Bayesian model, you'll of course have a prior over Z, the likelihood that tells you how likely it was to observe that D matrix from Z, and a posterior which tells you how much of certainty you have for having gotten that Z given that D. Okay, so now we look at the priors. The prior that we have over Z is the Indian buffet process, as it's called. It's a metaphor for the beta binomial process. I won't go into the details of this, but to just give you a mental picture, assume that you're at an Indian buffet process, Indian buffet, and you have like lots of dishes. There are n people in a queue, and the first person comes in, selects some of the dishes, goes and sits. The next person comes in, selects some more, again goes and sits. At the end of the day, if you try to compute the different customers versus the dishes they've taken, you would get some sort of a cluster matrix because everyone would have taken one of the dishes in common. And this is the Z matrix. The prior you put on that is known as an Indian buffet process. This might not be clear right now, but I'd be happy to talk into it even offline. This is Bayesian, therefore you don't have to infer the number of clusters ahead of time. The likelihood that we have here is a generalized Wishart distribution. We definitely had to tame and augment the vanilla Wishart so that it could handle distances. And this is, again, translation and rotation invariant. And then definitely, if you're a Bayesian, you have to end up doing some sort of sampling. So the model of choice in most of these IBP/Dirichlet process methods is MCMC. We have a Metropolis Hastings with a Gibbs sampler here. And what you do is you try to get approximations of your posterior distributions over time. And you're doing this for Z and the number of clusters. For anyone over here who's interested in how this entire thing works, this branch here is your prior. And this branch, or this setup here, is your likelihood. So if you invert this entire generative model on its head, that is, someone gives you D, the aim for you is to then find out what Z is. So that's the whole point of putting this here. I won't go into the equations right now, but I'll give you one example of where we tried this out. This is using the HIV protease inhibitors drug types that the FDA has approved. There are 10 of these guys. Here, this is the human cell, and you find the HIV is actually attacking. There are 5 different types of drug types that you can use to attack the HIV. It's always given in a concoction. And we've used the protease inhibitors that you see here. They are known to exhibit similar behavior because they have similar chemical structure, but they're not readily available. So this is actually a point of contention as to how to get the best drugs delivered at the right time, but you still have some anomaly in the drug space itself. So one of the ways is that we were trying to think is, can we find alternative PIs for therapy? And can we extract any structural dissimilarities that are present which are not already known? So what we do is we start building protein contact maps from these proteins. Each protein, as we know, is each protease inhibitor is a protein, meaning it has amino acids. So you compute the distances between these amino acids and build what is known as the normalized information distance through this. It's again a computational intensive step. And what you see over here, is the HIV reference genome built again as a contact map. And these are the contact maps of the other 10 protease inhibitors I've shown. Once we run POCD, the model of clustering, we get 3 different groups. The first group has ritonavir and atazanavir, so these 2 together, and they're normally administered together. The last 5 and these 3 here also fall into one group because they can be administered independently. And the one that stood out is atazanavir. This is so because it has structurally some differences which you couldn't really extract if you look at it, and it's also known to have lower cross-resistance. So this was one thing we could tease out from this, and our next experiments would be to put in all of the 26 drugs together and see what we would get. We also have another 2 experiments. One is on single-cell data, and another is on neonatal jaundice data, but this is unpublished data, and I unfortunately cannot show it here, but we're also writing this down. So this is my conclusion slide. I don't have a conclusion because this is work in progress. So if you still haven't understood anything from the talk, I'll just state 3 things that you can take away. 1, that it was a Bayesian model. 2, it allows you to cluster and find out overlapping clusters for objects that are present as distance data. And this, I thought I would share it to the crowd, which are actually good expositions about Bayesian non-parametric, so that's the first link. And this is a very good readout for the wars that you get between the frequentists and the Bayesians. This is the write-up about the paper I just explained, which was a spotlight in last year's NeurIPS. And this is a nice, cool place to find deep learning tutorials for awesome papers. And that's it. Thank you very much to WiDS for having all of us, as well as Swiss Re for hosting so many powerful minds. Thank you. And you can catch me here.
Hi. I have a question regarding the clustering. So
Sure.
when you say that you do the clustering, it's usually very computationally expensive, right?
Yeah.
And you do there like the pairwise similarity.
Yes.
So my first question is like, what is your sample size? What is you are doing in work in progress or whatever?
Yeah.
And the second is that if you have to select like a certain samples because it's gonna be a very expensive later on, what kind of sampling methods would you use?
Very good. So I had like these 2 questions. Someone would ask that, so I didn't tell it in the presentation. So okay, we have 3 experiments that we've run. For this HIV data set that I'd shown, even though we only have 10 drugs, once you build the entire strings and the distance matrices, they are like 20,000 times 20,000. So this is big enough, and it will make any MCMC sampler cry because you're actually wanting to identify, run through each of these in every Gibbs iteration. So the runtime complexity is O(n cube). I'll get to how we are planning to solve it. The second experiment is with single cell data, so that's like cells and genes. Again, 20,000 cells and 10,000 genes. So we want to identify which cells could belong to different genes, because it's always the case that one gene doesn't just express one particular, or doesn't belong to one signal pathway, it can belong to multiple. So you don't want to restrict that gene's contribution to just being in one cluster. So you want it to have in many different clusters. So this again did take a long time because we're still in the O(n cube space. And the third data set is that of neonatal jaundice cases. It's 140 patients from University of Basel, from the University Hospital Basel. And these are kids that have phototherapy treatment being given. So this is a small-ish data set, and this is where we first tried things out just to see if it worked. We knew the kids who had gotten treatment and who hadn't gotten, and thankfully the method did not just split it into 2. We have a mix of kids that have treatment versus non-treatment, but other features as well that tell us why they are different. So we will be discussing this as well. And now to answer how we are solving this, so if you just use a vanilla MCMC, Metropolis Hastings with a Gibbs sampler, you would probably need like maybe a week to get these things to finish and complete. We are using two things. We use an embarrassingly parallel MCMC. It was very hard to code that up. It's a beast. It's already a beast if you look at the equations. So embarrassingly parallel MCMC, this is already being done, and it's way faster. And the other thing we're trying is to do amortized inference. So rather than just do MCMC, we want to go into the VI space as well. And if you can do amortized VI, you can also get a deep learning kind of a formulation as well. So this is actually the work being done.
I have a sequence of super short questions. So
shoot it.
Like in the figures or like the results you showed, then every data point is one graph, right?
So which one? Do you want me to go to the slides or? Okay, let me just do this.
Yeah, like in this one, for example.
Yeah.
Like now every data point is one graph, so it's one molecule or protein?
This is, okay, so this is just the contact map. Now what you do is, so if you take, for example, let's say this one, it's 200 times 200. Yeah. We row vectorize this. So it's 200 times 200 vector. So you have 10 different vectors for each of these, and you compare it with the HIV reference genome here. So these are long-ish vectors, and you build a huge 20,000 times 20,000 matrix.
Okay, so because like my question is like my kind of question is, how do I represent my protein as a graph? So you have a sequence of amino acids, and now is the distance kind of like how many amino acids are between one?
Yes, it's the distance that's and the distance is computed using a normalized information distance here that you can see, which is actually a approximation of the normalized compression distance. I mean, we can talk about this offline. Okay. It is actually there. It's very theoretically in place, and it also uses the Kolmogorov complexity. So how would you move from, how much of effort would be needed to moving from A to B?
Because then it's hard to generalize to a graph where like one of the most important features is also like a connected or like a node vector associated to the graph.
Yeah, if you have embeddings that you if you have like latent embeddings, then it would be tricky to use that. So our formulation for now is things that we at least know. We still don't want to restrict ourselves to things that that may or may not have a latent embedding, but I understand your question. You just can't like plug things in, but we definitely have to give it a more thought. These are just using matrices that are huge, putting it in, because we know that these can't be directly embedded.
Okay, yeah, thanks a lot for your talk.